Hello, I'm Alexey Gurevich from St. Petersburg State University, and I would like to present you my poster about interactive visualizer of small molecule fragmentation in MS/MS data. Small molecules, also called natural products or secondary metabolites, are tiny but precious compounds for industry and medicine. For example, penicillin and many other antibiotics are small molecules. Currently, leading technique for studying small molecules is tandem mass spectrometry, but it generates a vast amount of data, and researchers use computational methods for analysis of this data. The state-of-the-art tools compare experimental mass spectra with large chemical databases of small molecules, and the tools model theoretical fragmentation of these molecules and compare so-called theoretical mass spectra to experimental ones. Unfortunately, these tools are not perfect yet, so manual validation always needed for their output. And for this purpose, we developed NPVs, a visualizer of mass spectrum fragmentation for a wide range of small molecules, because current proteomics visualization tools are not applicable to many types of small molecules. Here you may see a sample example of NPV's output. On the left, there is an experimental mass spectrum where each experimental peak matched to some fragment of the molecule shown on the right, colored blue, and the rest peaks are colored gray. A user may click on any colored peak to see how it corresponds to a fragment of the molecule in the database. There are many additional fields in the output which could be useful. For example, on the bottom, you may see a plot of accuracy of each explained peak and how it corresponds to the fragment with the size of the error in daltons. And also our tools support blind modifications where mass spectrum was matched to a new variant of a database molecule. In this case, the difference between molecule weight and total mass of the spectrum corresponds to a modification which happened to this database molecule, and NPVs detects the most likely location of this modification and colored it in orange. You may find many other examples of NPV's output at the project page. And note that this tool is currently work in progress, but we want to publicly release it very soon. At the moment, you may try it at GNPS platform, which is the largest community-driven resource for storing and analyzing small molecule mass spectrometry data. And our tool, our visualizer, is embedded into several popular computational methods at GNPS. So you may run it and find results and analyze them with our visualizer right now. I hope my flash talk was interesting and informative for you, and I would be happy to answer any questions at the poster session.